Good afternoon, everyone, and welcome to the next company presentation here at the Regenerative Medicine Investor Day being held in New York. I'm Robert Uhl from Westwick Partners, and I'm based in the San Diego office. Our next speaker up on the program is the CEO of Prima Biomed, so it's my pleasure to introduce Matt Lehman.
Well, thank you. It's a real pleasure to be here today. Really excited to be able to give you an update on Prima Biomed, what we're doing, and some of the highlights of what we've, let's say, some recent news and what's coming during the course of this year. So just briefly by way of introduction, Prima Biomed, we're an Australian-listed company. Our ordinary shares are listed on the ASX. We have an ADR, Level 2 ADR program, so we are fully reporting to the SEC. That trades on NASDAQ under the ticker symbol PBMD. So most recently, we're developing an autologous cell-based product to treat cancer. We reported towards the end of 2013 an early phase 2 trial in ovarian cancer. We're pretty pleased with what we've seen. We've been able to identify what we think is a target patient profile to move forward in clinical development. That was based on a small group of 20 patients. We're looking now, in the next trial will be starting imminently, so we've had approval from a number of countries, a number of ethics committees, a number of sites have now been opened in the last week or two. So we're looking at first patients coming in a larger randomized phase 2 trial, 210 patients. So I'll mention a few things about our clinical development program. And recently we did license the rights to CVAC to Neopharm Group, which is the second largest healthcare company in Israel. Has a pretty good network, especially for what we're doing, good knowledge base, good outreach to doctors. So that's for the territory of Israel and Palestine. That's our first license, commercial license deal for CVAC. So again, looking forward here over the next few months, and we're pretty excited about where we are and what we're going to take forward. I mentioned, like I said, the phase 2 ovarian cancer trial that's starting here probably early in the second quarter. We have a number of data catalysts out of that program in the ovarian setting, as well as we're starting a pilot pancreatic trial. So over the next few years, we have a number of interesting and I think very important data catalysts. Our cash position at the end of last quarter was 28.5 million Australian dollars. We also do benefit from some non-dilutive grant support, about almost 8 million euros worth of grant support in Germany. So these are research and development grants, as well as we actually benefit from there's a very nice research and development tax credit program in Australia. We receive have received about $1.5 million Australian dollars per year. So we just reported we got $1.6 million as a tax refund in Australia. So all in all, for what we're doing, we're in a pretty comfortable financial position currently. Just to talk a little bit more about what we're doing specifically, of course, in this conference, when we talk about regenerative medicine, cancer is certainly one part of that. A lot of people are excited about other therapeutic applications, but I think the idea of using cell-based therapy and how that fits into this very exciting field of cancer immunotherapy is especially valuable. And again, what we're doing and where that fits into that therapeutic field is really what we call the therapeutic vaccines. We're trying to stimulate T-cell responses, both active, cytotoxic T-cell responses, as well as facilitate a memory function in the immune system that can help control cancer over longer periods of time. So I think, on one hand, our technology is very much on this regenerative and cell therapy space. Tomorrow I get the pleasure of also attending cancer immunotherapy conference, which is more on the clinical side. And so, we're certainly a company that bridges both of those spaces. So this is briefly how we make our product. I mean, this is, we're taking white blood cells, monocytes, autologous monocytes from each patient. We bring them in, culture them, and separate these and allow these monocytes to start maturing into dendritic cells over 6 days. We then pulse this with a human fusion protein that contains mucin 1. And the idea here is we are, creating a product of highly sort of specific, highly purified cells, well characterized. We also think is important, very well formulated. So after the take-up of this antigen into the dendritic cells, we wash, concentrate, and fill and finish these as 1 ml vials. These are cryo-frozen, stable for, at least 18 months. That's the data we have to date. And so the idea is when we re-inject the product into the patient, these cells that have taken up the mucin 1. We're able to stimulate a T cell response, a killer T cell, as well as a helper T cell response that's able to target the cancer cells that have this overexpression of mucin 1. In mucin 1, it's a very interesting target for cancer immunotherapy. It's the version of mucin 1 that is present on cancer cells is very specific to cancer cells. We see this overexpression, and at least in our therapeutic areas of ovarian and pancreatic cancer, in the 80 to 85% range, so it's highly overexpressed in the patient groups of interest for us. This is from an NCI project that was run a few years ago looking at antigens for cancer vaccines. Mucin 1 was considered the second highest priority based on its therapeutic function, immunogenicity, functionality, those types of things. So just an overview here of our clinical trials. Of course, I don't have the time today to go over in great depth our entire clinical history. But basically where we are, like I said, we're starting a large randomized phase 2. To date, we're very comfortable with the safety and toxicity profile of this product. The toxicity or side effect profile we're seeing is very moderate, sort of immune-related reactions, injection site reactions, headaches, these types of things, which are expected with this type of therapy. None of them are very serious or severe, and they resolve themselves without further treatment. I think that's a very important thing for, especially when you're talking about cancer therapeutics, and especially as they get added on to other, cancer is usually treated with multiple combinations of therapies, and so the idea that you're bringing in something that's non-toxic is, I believe, very, very important. We're very happy with what we've seen in terms of the immune response. So we have some understanding of the mechanism of action. We've been able to verify the biological response. So we are seeing T cell responses. Those T cells are specific for mucin 1. We're able to test that. We've now seen this in a few different trials. And we do have some, so then in this trial, we have reported progression-free, or I should say top-line progression-free survival. We'll be looking at overall survival from this randomized trial towards the end of 14. This is a small extension trial, which we'll look at some kind of case studies and maybe some longer-term safety. This trial is there's 76 patients in first remission in this trial, so we'll be observing those patients over the next several years. So that's an extended trial looking at overall survival. And then these are the two trials we are starting in the second quarter here. So an ovarian cancer, 210 patient randomized trial, and then a pilot trial, single-arm pilot trial looking at resected pancreatic cancer. Here's a bit of an overview, again, of these trials and some of the data and different types of data points we have coming out over the next few years. But I'll speak here briefly about, of course, what's our most interesting and the most relevant, I believe, as our plans in ovarian cancer. So this is what was reported towards the end of 2013. We're having, I think, a very compelling clinical signal here in ovarian cancer. These are patients who basically went into remission after second-line based or platinum-based chemotherapy, which is very much the standard of care in this space. This is based on 20 patients. We're watching progression-free survival. We're seeing a 50% increase in median progression-free survival. I think also very telling, and if you do, let's say, watch the cancer immunotherapy space or understand a bit about how cancer immunotherapies work, we're also very encouraged by, let's say, the shape of these survival curves where, again, very typically early on you don't see a lot of difference. Around the median, we're seeing that 50% difference. And then the patients that are doing well, the blue, by the way, is the CVAC, and the orange is the control group. There's kind of this extended time in remission, basically, or time of progression-free survival. So we're very happy with the shape of this curve. It really makes sense. It really matches up with the logic of immune response and building an immune response. So as I mentioned, we'll be looking at overall survival. Well, we'll have our final PFS data set. That's coming relatively soon. Okay, I don't expect major differences from what we've seen here. But then we'll look at our overall survival is likely to be about the end of this year, end of 2014, when we expect the overall survival from this trial to be mature enough for analysis. Just brief, putting it in context, ovarian cancer, about 80 to 85% of patients do go on to second line. So ovarian cancer is an interesting cancer type because it is highly responsive to first-line surgery and chemo, but then there's frequently a relapse, or in almost all cases a relapse, and the vast majority of patients do go on to second-line therapy. if you look at that space, this is where most companies or most academics have been concentrating. The first line setting in ovarian cancer is a little bit difficult because of the heterogeneity of the patient population. We've experienced this ourselves. So in some of the trials we've run, we're not getting much of a read on clinical endpoints, either PFS or certainly not to date OS as well in that patient setting. So we will be looking at overall survival. As I mentioned, we have some of those groups and other trials ongoing, so we'll be looking at that over a number of years. But it's been that first line ovarian cancer space has been a real challenge for, I mean, the last company that gave it a real stab was Genentech with Avastin. They ran 2 very large trials, 1,200 patients each in a randomized setting. And, it's debatable what kind of clinical signal they were getting from that. So anyway, looking at the second line seems to be, again, based on our own clinical data, based on the literature out there, is really an optimal situation. We have a homogeneous patient population, that is where most patients do go into that setting. So we're looking at, this is an orphan indication, but it's a relatively, let's say on the larger side of an orphan indication. We're looking at, new incidence or a potential market for our, really specific target patient profile, about 25,000 patients per annum. That's in the major markets of Europe, in the US and Japan. And again, this is a little more specifically exactly where we're looking at bringing CVAC in. So right after the second line, platinum-based chemotherapy. And patients may or may not have surgery in that second line setting as well. So I won't spend too much time, but certainly this is a bit more of the detail on the immune response, the safety, some of the signals we were seeing in our phase 1 trial. Our sort of early pilot phase 2 trial, this was a single-arm 28-patient study, so we were starting to see some good signs of response using a tumor marker called CA125. And then this was the design of the trial that we reported progression-free survival. So again, it was basically 2 groups in this, first line and second line. And this was a randomized multi-center trial in the US and Australia. Again, the safety is really nice. The immune response is real nice. And again, these are the cell types and the sort of the cytokine expression that you want from your T cells. So we're very pleased with the types of immune responses we're seeing. This is as of September, so this is several months ago now, but we're looking at overall survival. We expect that to become mature. And this is a bit, as I mentioned, so this was the first line progression-free survival we reported. Again, this is something we'll be able to update here shortly as well as we continue to monitor the patients. The reality is in that first-line setting, there's a lot of statistical, I mean, we're seeing a lot of heterogeneity. Again, obviously relatively small numbers of patients, but it was very clear if we were going to get a signal in that spot, we'd have to be talking about very large trials and monitoring patients for very extended times. But what we are seeing in the second remission with a more homogeneous patient population is a nice signal, and I think we're pretty pleased, and this is what we're picking up with as we go forward. So a bit about what we learned. And clearly, as we're getting now in the later phases, this is where we'll be switching from, some of the earlier signals like CA125 and progression-free survival to overall survival as our primary endpoint. This is clearly what the regulators desire, and obviously the most clinically relevant and important to patients and doctors. So just a bit about this trial. Like I said, there's this first remission cohort, that was 76 randomized patients. And again, that is going to be a longer-term observational study at this point. So, we'll be monitoring those patients for overall survival. Second remission, this is the one that is now starting imminently. And then just briefly, a bit about we're looking at a very specific niche in this pilot trial of resected pancreatic cancer. And that's a 40-patient single-arm trial. And out of that, we are looking at how we can add in some potential additional indications for this product that's targeting mucin-1. And then we can make some go, no-go decisions on the back of this based on, how this looks against historic data and what let's say, the competitive situation is for cancer vaccines in this patient population as well. So. And then, of course, because we are at this regenerative meeting conference, I do want to mention a bit. We've done a lot over the last 3 or 4 years in terms of the actual manufacturing, the characterization of this product, everything from how we manage the supply and logistics, which is now automated with scheduling, labeling, how we ship these things. We have been able to transfer this technology into 3 different GMP sites, establish comparability, get the regulators to buy in on this. The quality control and the potency assays that we brought into development and now are validating. And again, what we're very proud of is that we've really formulated this into a product that is stable, small, easy to administer. And just a few looks at some of these things. So I'll leave you with that. I appreciate the time, and I believe there's some questions offsite or outside the room if you have any. Thank you.